Hello, it's my pleasure to participate in the International Brain and Biomedicine Conference, and I would like really to thank the organizers for this exciting meeting. I'm really sorry for not being able to participate in person, and I hope this will be on the next occasion. So today I will talk to you about how important nutrition is in motor neuron disease, also known as amyotrophic lateral sclerosis, ALS. I'm Luc Dupuis. I'm a basic scientist working at INSERM in the Strasbourg University in France. So what is amyotrophic lateral sclerosis, or ALS? ALS, it's a disease that is defined clinically. It's defined by the degeneration of both upper motor neurons, which are located in the motor cortex, and lower motor neurons in the brainstem and the spinal cord that degenerates together to define ALS. This combined degeneration of upper and lower motor neurons leads to progressive paralysis and death within 3 to 5 years after onset, usually at the age of 50 to 65 years. The lifetime risk of ALS is 1 in 400, meaning that 1 person out of 400 would die of ALS. So ALS is defined also pathologically. It's defined by the inclusion of one major protein, TDP-43, in more than 97% of ALS cases. The rest of the ALS cases are defined by inclusions of 2 proteins, either SOD1 or FUS. And ALS is a disease which has a sister disease, FTD, frontotemporal dementia. In this disease, more than half of FTD patients also develop either TDP-43 or FUS inclusions. What is important is that ALS is not only a disease of the central nervous system. It's also a systemic disease with a metabolic component. This is shown by 2 major features. First, ALS patients lose weight. This weight loss is caused by either decreased food intake or increased energy expenditure, and usually by both of these events. These 2 events are caused by different mechanisms, either dysphagia, altered hypothalamic signaling, or increased presymptomatic physical activity, which converge to trigger weight loss. And these metabolic abnormalities are also seen by the occurrence of systemic pathological metabolic abnormalities in tissues like pancreas, skeletal muscle, adipose tissue, or hypothalamus. But what is the cause of weight loss in ALS? Is it possible that the cause of weight loss in ALS is motor defects themselves? To address this question, we performed multiple studies, one of them being epidemiological cohort studies. You have here the result of one of this study. We interviewed ALS patients for their BMI changes before the onset of motor symptoms, and we also included a cohort of control patients. This is time 0, which is onset of symptoms. And as you can see here, ALS patients in continuous lines begin to lose weight more than 10 years before the onset of motor symptoms. This is also true in ALS mice. We have mouse models that lose weight before the onset of motor symptoms. Here are transgenic mice expressing mutant SOD1, and you can see that these mice lose weight here already, while they only develop motor symptoms several weeks later. So weight loss typically precedes motor symptoms also in mouse models. But are these weight loss also important or prognostic of ALS progression. This is a study from the group of Philippe Couratier in Limoges. In this study, they studied ALS patients either without 5% weight loss or with more than 5% weight loss. And what you can see is that ALS patients with more than 5% weight loss actually die earlier than the others. So this means that weight loss is a negative prognostic factor in ALS. But if it's a negative prognostic factor, could it also be a therapeutic target? And this we investigated first in mouse models. And to address this question, what we did was simply to modify nutrition of these transgenic mice. We gave them high-fat diet. By giving them high-fat diet, we were able to increase the body weight, and importantly, we were also able to increase their survival. So this means that high-calorie diet is able to increase the survival of ALS patients. If it's able to increase the survival of mouse model, is it also able to increase the survival of patients? To answer this question, we performed a randomized clinical trial in ALS patients in collaboration with the group of Ulm in Germany. And what we observe is on the general ALS population, no effect of the high-calorie fatty diet, here in blue. But when we separated ALS patients with fast progression, which are more than half of ALS patients, as compared with slow progression, one can see that the patients with the high-calorie diet supplementation live actually longer than the placebo-treated patients. So this means that, at least in the most severe forms of ALS, nutrition might be critical in driving disease progression. So what are the first conclusions? First, ALS progression is related to weight loss. Second, targeting weight loss is able to improve disease progression. But the question is now, what drives weight loss? What is the neuroanatomical substrate of weight loss? And then we have to identify the possible brain region that might be driving weight loss. And one obvious candidate is hypothalamus. Why hypothalamus? Because this is the brain region that is setting up the interface between metabolism and in particular food intake or energy expenditure with the peripheral organs, including liver, muscle, or stomach, adipose tissue. So all of the signals that are coming from these peripheral organs are integrated in the hypothalamus, leading to control of food intake and energy expenditure. So is the hypothalamus abnormal in ALS? Well, we have made one study in ALS patients which was based on magnetic resonance imaging, and we quantitated the volume of the hypothalamus in a cohort of healthy control, here in black, ALS patients, here in gray, but also familial ALS patients, and most importantly, presymptomatic familial ALS gene carriers. So this last group are people that are not yet sick, while they have a 100% risk of developing ALS in the 10 to 20 years because they carry an ALS-associated mutation. So what do we observe in terms of hypothalamic volume? We observe that healthy control have a larger hypothalamus than all of the three disease groups. So this means that hypothalamus is atrophied in ALS. Is it related to weight loss? Well, actually, correlation analysis showed that there was a weak correlation between the hypothalamic volume and the BMI in ALS patients, but this was much stronger when considering only familial ALS patients. So if the hypothalamus is somehow sick in patients, what part of the hypothalamus is actually driving weight loss? This is a complicated question because there are more than 50 different hypothalamic nuclei, and many of them are actually involved in weight loss and energy metabolism. So what we did was to study pathologically hypothalamus in patients with my colleague Dennis Yilmazer-Hanke. And what Dennis found was that the LHA, the lateral hypothalamic area, is the region where there is a concentration of neurodegeneration, as seen here with lipofuscin, and also of TDP-43 inclusion, as shown here. So the hypothalamus is not uniformly atrophied. There are regions, and especially the LHA, which are more affected. In the LHA, there are several neuronal types which are well known to control weight loss and energy expenditure. One of them are neurons which express a neuropeptide which is called MCH, melanin concentrating hormone. And these MCH neurons we observe are degenerating in ALS patients. You see here an MCH neurons from control patients, and here from an ALS patients. You can see degeneration of these neurons. And what is more important is that this loss of MCH neurons is also observed in ALS mice, either SOD1 or mutant mice. What could be the consequence of this loss of MCH neurons? We investigated this by complementing our transgenic mice with MCH. So it's kind of a heroic experiment where we implant in the brain of these mice a cannula, and this cannula is connected to an osmotic mini pump that delivers the peptide directly in the brain. And once we do this, we let the mouse recover for a few days, and then we record their metabolism. What we saw was that providing mice with MCH was able to slow down their weight loss, and you can see that the purple mice are untreated ALS mice, and when they are treated in orange, you can see how they regain weight. And this is due to increased food intake because these treated mice actually ate much more than the untreated mice. So loss of MCH blunts food intake in ALS mice. As a conclusion, what we show in total is that ALS progression is related to weight loss, that when we target weight loss, we improve disease progression in at least a subset of patients, and that this is most probably due to hypothalamic defects, as hypothalamus is structurally and functionally altered in ALS, and because hypothalamic neurons orchestrate at least some of the metabolic abnormalities. The questions that are currently ongoing in my lab as whether there could be other consequences of hypothalamic alterations, and in particular the neurons that we identified as being very affected in ALS are also involved in sleep. So maybe sleep is altered in ALS patients, which we could treat. Last but not least, we would like to target metabolism and weight loss to improve progression, but with a pill, without dietary interventions, which are somehow difficult to reconcile. So this whole work has been done in a tight collaboration with my long-standing friend and collaborator, Professor Albert Ludolf in Ulm, Germany, and many colleagues in Ulm, many PIs in Ulm are involved, in particular Professor Kasubek for the MRI, Professor Yilmazer-Hanke for the pathology, Professor Roseli for many mouse studies, and Professor Nagel for all the epidemiology studies. And in general, this work was performed in my lab in Strasbourg, in our newly built CRBS, Center for Research in Biomedicine of Strasbourg. And the two important people in the lab on this topic are Mattei Bolborea on the right and Simon Guyot in the middle, circled in red. I would like to thank you for your attention and to apologize once again for not being able to be there with you in presence. I hope that at some point in the next years I'll be able to visit Pakistan to visit your labs and university, and I would be really glad to do that. Thank you for your attention.